I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of lecture of forensic science, we are discussing various tools and techniques used for analysis in various divisions of forensic sciences. In our series previously we have discussed the tools and techniques used in the ballistics division, in the documents division, in the fingerprints division. Today we will talk about various tools and techniques or the instruments which can be used in the biology division or the forensic psychology division. So firstly we have to look what nature of evidences are being encountered in the biology divisions. Various biological specimens are being encountered in the biology divisions. For example, the specimens from our body, the secretions from our body like the blood, saliva, tears, semen, urine, etc. These are the biological samples which are collected from the human body and which are being analyzed in the biology division of the forensic science. Apart from that, we have various samples of insects, pollens, etc. like the forensic entomology or the forensic palynology. So those samples are also being encountered and being analyzed in this biology division. There are various, before going for the analysis, like we look for the class characteristics and the individual characteristics for the identification of any evidence. In such cases, we look for the preliminary test as well as the confirmatory test. Let's suppose we have a sample of blood and the crime scene. So what would be the first question? Whether the substance which is present at the crime scene, is it blood or not? Because sometimes that could be a fake crime scene and instead of blood that could be a chemical substance. So first of all we have to see whether the substance which is present at the crime scene is blood or not. After confirming with the preliminary test that it is blood, we have to look for whose blood it is. It could be any animal's blood or it could be human blood. So we have to see the difference whether it belongs to a human or it belongs to an animal. We call it the species identification. After confirming that it belongs to human, we have to identify whose blood it is. So we have to look for that this blood or this DNA belongs to whom. So for the first type of preliminary examinations, we have certain microscopes. The major microscope which is used in the biology division is a compound microscope. A variety of compound microscopes are available to the forensic microscopist and their selection will depend on the types of evidence which is to be studied. For example, the bright field microscope is there, phase contrast microscope for a contrast sample, comparison microscope if we have to compare two evidences together, hot stage microscope, fluorescence microscope in the cases where the evidence emits some bioluminescence or fluorescence, and the polarizing microscope for the evidences which are polar in nature. So the evidences for the preliminary examination are first examined under a compound microscope and we look for pollens in the cases of various pollen examination or the insects or the hair, fiber, etc. under the microscope for the preliminary examination. So as we discussed, the nature of evidences which can be found and which goes to the biological division include blood, semen, saliva, sweat, hair, bone, urine, feces, animal materials, insects, bacteria, etc. All these evidences goes to biological division for analysis. So firstly, we have to look whether the substance is blood or not. We can look under the microscope and see, yes, the RBCs are present, but intact RBCs are present only for certain hours. The RBCs of human and animals are different. On those basis, we can clearly identify that this substance is blood and is this a human blood or an animal blood. After this confirmation, we go To the next part, to confirmatory test of the blood examination, we can use Stickman test, Takayama test or Wegener test, which these three tests, any test can be used for the confirmation of the blood, and the crystal, depending on what kind of crystals are being formed, we can confirm that this substance is blood. This is a confirmatory test for the presence of blood. Another technique which we use to know the derivatives of blood, we can use for the UV spectra. Different components give different spectra at different wavelengths, and those wavelengths can be used for analyzing it. So after the confirmation that this substance is blood and this is human blood, we go for the DNA extraction, because through DNA extraction we can find out that this blood, if it is found at the crime scene, whose blood it is. Is it victim's blood? Is it the suspect's blood? Is it the criminal's blood, or any other person who was present at the crime scene, whose blood it is. So after the proper extraction of DNA, depending on what kind of specimen you have got from the crime scene, whether it is blood, it is semen, it is saliva, it is nails, any vaginal swab, etc. We use the appropriate technique for the extraction of DNA from these specimens. After the successful extraction of DNA from these specimens, we have to make number of copies. For that, another major technique we use in the biology division is the polymerase chain reaction, also known as PCR. Since this technique was being founded in the Carey Mullis in the year 1984, the work of molecular biology has reached its heights. The PCR is a technical method in molecular biology to intensify a solitary or some copies of part of DNA, producing millions of reproductions of a definite DNA sequence. It is applied in forensic research laboratory and is beneficial since a small quantity of original DNA is mandatory and we can make copies out of it. The forensic cases or the criminal cases are such cases where the evidence is limited. Whatever evidence we have found at the crime scene, it is very limited and it cannot be replicated, and we cannot procure that evidence again from any place. So we have to make the maximum use of the evidences which are being found at the crime scene. Whatever DNA we have extracted from the material found from the crime scene, we will PCR and we will make the copy number of DNA. So in the PCR cycle, we have 3 parts where we can make the copies of a particular DNA sequence using the specific primers for particular gene. After making million of number of copies, we have to sequence this in the form of ATCG. The sequence will come, and then we can match that this particular DNA sequence matches with this person. For the matching of the DNA sequence, we have certain techniques. Either we can go for RFLP, VNTRs or STR analysis. Restriction fragment length polymorphism method is a technique which may be distinguished by analysis of patterns derived from cleavage of their DNA. So we use particular enzymes, and the enzymes will cut DNA at particular places. And then we have short segments called VNTRs, variable number of tender repeats. The VNTRs and the occurrence of VNTRs in different persons is different, and how frequently those VNTRs are appearing in the DNA of that person will help us in identifying the suspect or identifying the criminal from the DNA sequence which we have retrieved from the biological specimen which was present at the crime scene. So with the DNA extraction, we first PCR with the labeled primers, and then we digest with restriction enzymes, The restriction enzymes like EcoR1 etc., they digest the particular DNA sequence at specific sites, and then we electrophoresis it. After electrophoresis, we can get the various strands of DNA of various length. For example, 200 base pair, 300 base pair, 350 base pair, and with the laser detection, we can identify the perfect amount or the perfect section we were looking for, whether this shows a match or not. If two entities diverge in the space between position of cleavage of a specific restriction endonuclease, the dimension of the fragments formed will differ when the DNA is processed with the restriction enzyme. The resemblance of the arrangement produced can be applied to distinguish one species from another species. The gold standard, which is being used in forensic science for the DNA matching, is short tandem repeat. When we were talking about the VNTRs or the variable number of tandem repeats, the variable number of tandem repeats is a big sequence, but short tandem repeats are a sequence of 4 to 12 base pairs. So we have in forensic the major shortcome we can say is the sample which is being procured from the crime scene. So as the sample is limited, and we can sometimes the sample has it is in a form that it cannot be the DNA which we have extracted from the sample, the only STRs can be found of those particular sequences. We cannot go for the long repeats. In such cases, we look for the short tandem repeats because DNA is degraded and we have very less amount of continuous DNA present. So STRs, we have around 4 to 8 or 4 to 12 repeats, and we look for this repeats in the person, and that can be further matched. DNA areas with the short repeated units are known as short tandem repeats. Those remain selected for forensic use usually have a tandem repeat unit of only 3 to 4 base pairs, and they may be repetitive in the DNA molecule from a few to dozen of times. So in STRs, we first go for the PCR with the primer 1 and the primer 2. After the PCR, we go for the electrophoresis, and then we look for the fragment size, that how many times that particular unit is being repeated. Let's say, for example, we have identified the STR unit AACC to be a pattern in a person. So AACC is repeated in me 18 times, but in another person it is repeated 25 times. So that repetition is different. If my blood is found at the crime scene, if it matches, then it should show a repetition of 18 times. So that's how we identify any blood sample or any DNA sample and matches using the number of times that particular repeat is being repeated in that person. This can also be used in the analysis of cases of parents, paternity testing, that number of repeats, it is similar in the parents, both the parents, as we acquire DNA, 50% DNA from the father, 50% DNA from the mother. So we have the number of repeats similar to them. The major advantage of this method is that only minor quantities is required even in severely degraded DNA, which may be sufficient for forensic use. The difficulty is that a very minor tester of very small DNA segments, the short tandem repeats, require to be amplified in size for investigative accessibility and efficiency. So with the DNA extraction, we go for the multiplex PCR and then the capillary electrophoresis. We go for the data analysis, we have the repeats, and then we look for the particular locus, and then we identify the cluster STR search. After identifying, we can give 100% conclusive result, excluding the twins, that this particular DNA belongs to this particular person, DNA is one such evidence which help us in the personal identification of a person. As we were talking about the fingerprints, fingerprints are the evidence which cannot be even similar for two twins, but DNA can be similar for the monozygotic twins. So in such cases, except for the twins, the STR matching or the DNA matching is never same. So whatever report we'll procure out of this matching will show us a conclusive result of the personal identification of a person. So next samples or the next division where we have is the chemistry and toxicology division. The various samples which go for the analysis in the chemistry and toxicology division are the poisons, paint samples, drugs, or we have certain inks analysis. So all the chemical compounds which are being analyzed, they go to the chemistry or toxicology division. After, if there is a case of unknown death, in the autopsy, the viscera which is being collected from the person's body, after the autopsy, the forensic pathologist, he collect the various organs like a part of kidney, the part of heart, or the contents of stomach. All those viscera or the various body parts, they will go to the chemistry and toxicology division to know, in the cases of unknown death, whether the person has died consuming some poison. So various tests are being conducted in such cases to firstly know whether any poison is being present in the person's body, and if any poison is present, then which poison has been given to the person, has been given to the victim for this particular killing. Various techniques are being used as we talk about the preliminary and confirmatory examination in the cases of biology division. Similarly, we go for certain preliminary examination in the cases of chemistry and toxicology division to know whether the substance which is being which is being sent by the forensic pathologist, that contains poison, and what kind of substance is that. And after the confirmation, we go for the confirmatory techniques like spectroscopy, microscopy, etc. Spectroscopy and chromatography are two techniques which can give us the confirmatory result of which kind of evidence is being analyzed. Spectroscopy methods are very rapid and they are very less time consuming. They give information which is recorded in the form of a permanent chart, and in the form of peaks we can get the structural information which is being gained by the spectroscopic methods, and we have the precise results and reliable results where we can identify two different compounds. Supposedly, if we have received, or if the chemistry and toxicology division, they have received a white powder, they have to identify whether this white powder is a drug or not. If it is drug, which category of drug it is. So by looking at the color, by looking at the texture, and by not tasting it, because we don't know what kind of substance is that. So we just look at the color, at the texture, and we reach to a certain decision that it could be this drug, A drug or B drug, depending on its color and depending upon its texture. Then we go for some color test, we put some chemicals and we see, okay, this is showing positive for this particular drug, but we have to identify that what are the components of this white powder. Then we go for the spectroscopy techniques or the chromatographic techniques to know the structural information of that particular compound, that this compound has what kind of chemicals present inside it. So we will go for the atomic spectroscopy and molecular spectroscopy, where in the atomic spectroscopy, where the atoms are in the ground state, a particular amount of energy is being provided, and after receiving that particular amount of energy, the atoms from the ground state, they go to the E2 state. That means they receive the energy and they go to the excited state. After going to the excited state, they release certain energy. That energy which is being released after going to the excited state, that is the fingerprint energy of that particular component. This released energy comes to us in the form of spectra, and we identify that this particular group, chemical group, is present in this particular compound. We can see the spectra in the form, and we can say that we have a CH2 group present, CO group present, CH2 peak is there. So depending on what kind of chemical group is present, we have a different group at a different frequency. For example, a asymmetric stretch of CH2 group gives us a peak in 2850 centimeter inverse. So we have a database of all these peaks of different drugs, of different poisons, of different paints, and from this database we can match that this particular compound matches with this, and this is this particular drug or paint sample or poison sample. The spectroscopy which are being used in the division of chemistry and toxicology is infrared spectroscopy, which can be used to identify the functional group presence in the organic compound. So it is helpful in determining the chemical composition of substances like drugs, explosives, fibers and paints. Then we have UV visible spectroscopy, which is useful for detecting and characterizing organic compounds, and particularly those who have conjugated systems. Mostly in UV visible spectroscopy, we go for the colored compounds. If you have certain ink for analysis, some dyes are there, some pigments are there, or some colorants which are found in the fiber, paints and inks. The UV visible spectroscopy is also being used for the analysis of blood. We can also get to know that how old is blood which is found at the crime scene. In the cases of fiber paint examination, we can get peaks of different components which are being added in these substances, and they give us different peaks at different areas. In the cases of ink analysis, from the question document section, sometimes we get a blob of ink. So we have to identify whether these two ink matches with each other or they do not. Another spectroscopy which is being used is nuclear magnetic resonance spectroscopy. We call it NMR spectroscopy. This provides the molecular structure and composition of organic compounds, and thus it is often employed to analyze various drug, toxic substances and other organic substances which are encountered in forensic investigation. Apart from spectroscopy, we have a major technique, chromatography. This is basically, we have the ingredients are in a combination and they are not chemically combined. So consequently, they can be detached over some physical procedure. So we go for, in the chromatography technique, we sort out the constituents or solutes of a combination on the basis of comparative quantities of each solute dispersed between a moving fluid stream and a continuous or the stationary stream. So for the chromatography, we have a stationary phase and a mobile phase which can be used to distinguish, or you can say sort out, the different components. For example, if we analyze any ink sample using TLC, that is thin layer chromatography, so if we put a blob of ink on the stationary phase and the mobile phase will carry it upwards, it will diffuse into 2-3 parts depending on the chemical composition of that particular part. As you can see in the image that we have started with the ink spot, and that is basically the stationary phase where we have put the ink, and the water which is present inside the beaker, it works out as a mobile phase, and the components of ink are beautifully distributed evenly on the stationary phase, depicting the various components or the pigments which are present to form the ink. So with TLC, another important technique is gas chromatography. This is used to separate volatile compounds. This is a confirmatory technique which is commonly used to analyze volatile organic compounds which are found in ignitable liquids, drugs and toxic substances. Gas chromatography can be used to analyze volatile substances like the petrol, kerosene, etc. So it's most commonly used, or alcohol, etc. Most commonly used in arson cases. Liquid chromatography, we have HPLC and thin layer chromatography, high performance or high pressure liquid chromatography, which are used to separate and analyze a wide range of compounds which includes drugs, poisons, dyes and fibers. Apart from that, we have capillary electrophoresis, which is a technique that separates charged molecule based on their electrophoretic mobility. It is valuable in the analysis of DNA fragments, amino acids, drugs and other charged substances found in forensic samples. So in the electrophoresis techniques, these are the confirmatory techniques which give us the confirmatory results. Depending upon what kind of sample we are analyzing, we have to choose our technique. For example, the gas chromatography is used for the substances which are volatile in nature. For the liquid or the liquid samples or the solid samples, either we have to dilute those samples and we can go for TLC, or we can go for the HPLC of those samples. For the drug cases, we use narcotics identification kit. This system of narcotics is the only global customary for field testing apprehensive substances. This is the most widely used and successful presumptive testing system in the market today. And investigative laws and experience knowledge of the characteristic of suspected narcotics and the expected color reactions from the field test to appropriately classify assumed narcotics. The narcotic identification system is designed to presumptively identify compounds based on probabilities and the use of multiple tests in succession. The more tests used in the identification of a substance, the greater the possibility of a positive indication. So the drug identification system will go the drugs or the poisons, they will be identified in the toxicology division. For the forensic chemistry division, we have the paints, the dyes, the pigments related substances and various other volatile compounds which are being identified in the cases in the forensic and toxicology divisions. These forensic techniques contribute to the crime prevention and the development of investigative strategies. By analyzing patterns in crime scenes, behaviors or modus operandi, forensic experts can help law enforcement agencies identify potential serial offenders, predict future criminal activities and establish proactive measures to prevent such crimes. Overall, forensic techniques are vital in gathering, analyzing and interpreting evidence in a systematic and scientific manner. They provide critical information that helps in solving crimes, supporting legal proceedings and ensuring justice is served. Thank you so much.